Hello! Welcome to
Hi there.
Microbiology. Welcome to YouTube Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicating the public with the tools of biotech.
My name is Faz, and in a previous life, I got my PhD in microbiology from Imperial College London, where, amongst other things, I was making the flesh-eating disease glow in the dark. And I've also worked as a research integrity specialist, and these days, I'm an editor for an academic journal.
Every week, we meet to talk about microbiology, and today's our deep dive week, where we look at each figure in a paper that we chose last week to learn and criticize the scientific findings. This is a journal club, and we encourage our audience to leave us questions and comments.
Next week will be our news week, where we survey a bunch of different articles to choose one to do for the deep dive after that. So make sure to subscribe to satisfy your microbiology curiosities.
You can follow along with the papers that we discuss on either week in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use comments or tweet us using the microtwjc hashtag.
And so this week, the paper that we chose was Pore-forming Esx proteins mediate toxin secretion by Mycobacterium tuberculosis.
Yeah. So, Mycobacterium tuberculosis, you may know this bacteria from such diseases as tuberculosis. And it's
But that
it's
family
a
causes also some other like really terrible diseases, right? The Mycobacteria family, there's a lot of, we'll say like long term, right? Like I think a good thing that characterizes its disease forms is like long lasting. So leprosy is
yes.
also from the Mycobacterium family.
Yeah. So yeah.
Yeah, so I
So
pulled out this slide from a review just to go over some of the basics of tuberculosis disease. Maybe another thing people might not, or people might be aware of, it's like antibiotic resistant is a big problem in Mycobacterium. There's lots of bad things we can say about Mycobacterium. The disease itself it's a lung disease, and what's really interesting about it, and what it was always framed to me when I was going through school was that it's really fascinating because the immune system plays this huge role in it. Where
yeah.
like the macrophages that eat up bacteria, they eat up this mycobacterium, but then the mycobacterium doesn't really die, and it just like recruits even more macrophages to come in and kind of cluster around the infected macrophages until you get like this big blob, a structure called a granuloma.
Mm. Yeah, it's fascinating because tuberculosis basically turns your immune system against you, but in a very different way to say other diseases like it's just like the moment like a macrophage engulfs a tuberculosis and it's about to kill it. Tuberculosis is like, no, no, this is what I want. I'm
Yeah,
gonna
exactly.
take you over now.
Yeah, it wants to be inside of a macrophage. It wants to get engulfed, and it makes its lifestyle is basically predicated on that fact.
Yeah, it's
Um,
like the Joker where getting caught is part of its plan.
yeah,
It's just
that's right. I like that analogy.
it's just inside the little vacuole and going, well done.
Okay, let's look at the next slide I
Yeah,
got.
next slide. So going into like how does that happen? Does tuberculosis turn these macrophages against themselves? Um,
Mhm. With
so yeah,
secreted proteins.
yeah, secreted protein. So so
Yeah.
it manufactures these proteins that usually virulence factors that work to do all sorts of things to mess up the immune system, and they
Mhm.
secrete them using. Well, so I mean, do you want to talk about these various secretion systems?
Yeah, absolutely. So, like, I think it's like a commonality, right? Like, if you want to affect the outside world, you got to get things that you're making on the inside to the outside. And so, yeah, Mycobacterium, it wants to hijack, right? Like your macrophages. So it has to secrete things from the inside to the outside. People have been studying secretion systems in bacteria for like a huge amount of time. And they've generally come to a classification system for them as they've discovered more. So, like, there's these six different types. Mycobacterium, it has like a slightly different structure of its cell envelope. It has this outer membrane that's made of really unique compounds, waxy, waxy. I guess that's the best way to
Yeah.
describe it. Like kind of a waxy outer coating. But like fundamentally, it's still this idea that there's sort of two layers. There's like an inner membrane and an outer membrane. So in the classification scheme of things, Mycobacterium secretion systems got tagged on to the end, and they're called type 7 in general.
Yeah, and these act like molecular machines that pump out whatever these secretion systems particularly want to pump out.
Yeah,
Um,
there's all sorts of ways that they do that, right? Like sometimes you just pump things into the middle space, the periplasm, and then another protein takes over and pumps things to the outer, right? Or sometimes it's like a tube that goes straight from the outside or straight from the inside to the outside, and sometimes that tube even goes all the way across like and injects things into another cell.
yeah, so
Um,
like some of them like are directly injecting into the host membrane up top, and uh, it seems like Type 7 don't quite do that, but they just pump stuff into the just where into a very confined space, which is inside the vacuole that the phagosome uses to engulf them.
mhm.
So yeah, so when a phagocyte engulfs something, it puts them in a little pocket inside of itself to
Yep.
digest things, almost like a temporary stomach. And the mycobacteria turn that temporary stomach into a nice little home for the mycobacterium to live in.
Yeah. Yeah. So secretion is important in this sort of phase. And so like just to give some context about mycobacterium and like what's known about this secretion system or how it came to be known. So you guys in the UK, you guys have this vaccine for
Yeah,
mycobacterium, right?
BCG.
BCG.
Yeah,
Oh, do you have the scar? You're gonna show
I
us the
still I still
scar?
have my scar. I was about to roll my sleeve, but then I realised this jumper's too much. So I'm not sure whether I can show off. But I think I've got it over here.
Oh yeah, I see
Yeah,
something. Yeah, I see a dark
I've got
spot.
my
Hmm.
BCG scar.
So this image is from a baby, so it looks a lot fresher, right?
Yeah.
But that scar is indicative of getting this BCG vaccine. It's a inactivated or attenuated strain is one of the words that we use to describe it, meaning that it doesn't cause serious disease,
Mm.
but it is a live bacterium. Or maybe no, I think they heat kill it, right? It's heat killed.
Yeah,
Yeah.
but
So
yeah.
it's dead, but it comes from like a living thing that they killed. And it doesn't, it has, it can't make serious disease. That's one of the reasons why they use it is because in experiments they found, oh, it doesn't actually make that serious of disease if it was alive anyways.
Hmm.
And an important difference. So like this is I mean this is great. I love talking about this in light of vaccines because you make vaccines, you don't always know the mechanism on how it works. It works, you don't know the mechanism, right?
Hmm.
And so people were like, why does this strain not cause as serious of disease as like the fully virulent form? People didn't know, right? And so what they did was they compared genetics, and they're like, oh, there's all these regions of differences, right?
Hmm.
And one of these regions of difference created a secreted protein called ESAT6. So like that C figure is like showing this region of difference one. There's all these proteins in it. And then that D figure shows that they blotted for this particular protein ESAT6. And if you didn't have the region of difference, you didn't secrete ESAT6. And if you did have that region of difference, if you even put it back into things that don't normally make that secreted protein, it was able to secrete this again.
Right.
So this is like one of the classic virulence factors.
Yes.
And what was found is that in that same locus of genes, there were also other genes that if you knocked it out, they weren't able to secrete either. So it wasn't just that like this protein could get out by itself, but there was something in this cluster of genes that like helped things secrete. And so here's some experiments from that paper. They like complemented things back. It was like a cell culture experiment to see whether or not the cells died,
Yeah.
basically, or the cells like had worse metabolism. And then they also did it in mice and to look at abscesses in mice.
Yeah. So basically, this is what we'd call a virulence factor, where
Hmm.
there is something where it benefits the bacteria, but it's not always clear exactly how they work, because that's still something that we're trying to figure out. All these things are quite
Yeah.
complicated.
Yeah, no, I think that that's why I want to tell this story because in a way, the story that we're going to talk about is how do you find out like what these things do, right?
Yeah.
You know they do something, right? Like you have this evidence that oh yeah, in disease it messes it up when you remove it. But what is it doing in disease that makes it, right? What is it doing? Like that all the science that we've just looked at, I mean, four figures or whatever, it
Mm.
doesn't tell you what it's doing, right? It shows you that like when you remove it, it doesn't do the thing, but like what is the thing doing? And
Yeah.
that's like the big challenge, I think, of like understanding biology, really.
Yeah, because we do a lot of knockouts and stuff to find out the what, but the how takes a bit more time.
Hmm.
And this
Hmm.
paper is going deep into the how.
Yeah,
Um,
trying to.
yeah.
Yeah.
So yeah.
So lots of experiments have been done since that time. Sorry, did you want to take
Oh
this?
no, no, you go for it. You're on a
Oh,
roll.
lots of experiments have been done since then, right? And people have knocked out all these different components. Like, they have this whole region of difference to look at, and they find, oh my gosh, there's like related regions to this all over the Mycobacterium genome, also all over bacteria in general.
Yeah.
Like staph, I think I may have mentioned this when we chose this paper. The reason I know about type 7 is because I studied Staph aureus, and Staph aureus has a type 7 like secretion system.
Mm.
And so this is a kind of an interesting thing that when you find something in bacteria, then you start comparing it along all these other bacteria. You're like, oh my God, it's everywhere.
Yeah.
And this is one of the more recent secretion systems that have been discovered. If you see some of the paper dates here, they're all like in the 2000s.
Yeah.
And so yeah, so there's a secretion system. They know it exists. They actually don't really know what does the outer membrane part of the secretion system. So
Mm.
like through all their different knockout experiments, they know that there's definitely this inner membrane ATP dependent mechanism, right, to export the proteins, but they don't really know how to get from the middle space between the inner membrane and the outer membrane out into the outside.
Right.
That's a bit less clear.
So the structure of these proteins, these proteins all have this WXG motif, right? Which is
Mm-hmm.
I can't remember the exact amino acids that represents, but
Tryptophan,
trips.
and X is anything, and then the G I think is glycine. But don't
Right.
quote me on those.
Yeah, I think glycine is right. Tryptophan. Yeah, that makes sense somewhat, I guess. But yeah, I think that. But the point is,
Yeah,
this is like this consensus sequence for proteins that are about 100 amino acids long, so they're called the WXG 100 family, I guess.
yeah, and that's the one thing that they find everywhere. That's actually the main way in which these genetic loci are characterized, right? It's
Mm.
like you got a couple of WXG motifs, you have something in the inner membrane that exports things. That's a putative type 7 secretion locus. And confusingly, because we're in the world of genetics and sort of scientific mumbo jumbo, type 7 secretion may also be called ESX.
Right.
And then
Yeah.
so when they name these proteins, they're all like ESX, a letter or a number, but that more or less corresponds to some sort of type 7 secretion substrate or mechanism. Unsure, right?
Right. Yeah. So this we're kind of very much in the anachronistic territory of different enzymes have many different names, but I think the thing you need to focus on is that these we're going to call refer to for the next for all the rest is ESXs.
Yes, ESX, that's right.
Yeah, and so this lab that we're going to be looking at, they discovered this CPNT protein which creates channels in the outer membrane which allow it to kind of I guess grow, but also like the interesting thing is that a part of it has like cytotoxic effects, and I think it snips off and floats around and can mess with macrophages and actually
Yeah.
cause toxic effects on them in cell culture.
Yeah, I love that. Like, I think we've been talking so much about viruses in this past time, and I don't know if our viewers are up on it, but viruses have this great way where everything gets compressed in the genome, everything's multifunctional, right?
Mm.
But even in bacteria, like proteins, there's a lot of evolutionary pressure for proteins to do multiple things, and that's going to be true in eukaryotes as well. So like, here's a protein that they have, a putative virulence factor, right? Because it messes with macrophages, but it also lets them grow better. Like in the studies, that like they intake better glycerol and other nutrients into their bodies near the cells.
Yeah, I think the C-terminal domain of CPNT is quite interesting. They're calling it tuberculosis necrotizing toxin, and what it does is it hydrolyzes
Mm-hmm.
an important like kind of cellular factor called NAD, which takes part in cellular respiration, helps to do a lot of important things for the cell to ensure it has enough energy. And so having
Yeah.
this factor floating around the cytoplasm of the macrophage causes it to die, and that's not a good thing for your macrophages. Um,
Yes. Yeah. And so, like, I mean, this gets real complicated, right? Because, well, like, there's so many things that get secreted. If you imagine like living inside of an organelle that's
mm,
supposed to be killing you,
mm,
that's like what Mycobacterium wants to do. Like it has to control so many elements, right, of that scenario. And so, I mean, eventually it does want to kill the cell and get out of there so it can spread. So, yeah, this is thought to be maybe one of the keys to that killing phase.
Yeah, I mean, it's an agent of chaos. It's just going to
Yeah.
co- disrupt the
We're
cell.
going back to the Joker
Yep, going back
analogy.
to the Joker analogy. Tuberculosis is the Joker. Quote me on that.
Okay, so let's dive into the paper, I
Yeah, so
think.
we know that this toxin is produced, we know that it's excreted, but we don't know how it does that, and that's what this paper
Yeah.
is aiming to delve into.
Yeah.
Uh,
So looking in that genetic region where CPNT is made, there are these two ESX proteins that are there. And so, hey,
yeah,
ESX proteins, we think they're involved in secretion. Maybe these are the things that we should investigate to figure out if they're doing anything. And another important part of this whole puzzle, in terms of like just what this part of the genome does, is the IFT locus. So, CPNT, it has this toxic activity on NAD. Well, actually, bacteria also have NAD. So, like, IFT is the antitoxin that they use to make sure that this toxin doesn't get prematurely activated and go crazy inside of its own cell to kill it.
yeah. I mean, it's an important thing to not be allergic to your own venom. I mean, like many
Hmm.
venomous animals, the Mycobacterium have to make sure that. So this IFT it binds to the CPN. I think the thing that causes the necrosis to stop it from
Hmm.
acting on NAD. So I think so. It's a very important factor in this. Um,
Yeah.
yeah,
And so here they have this is the operon that you can see in figure A. That's the operon meaning the region of DNA. And then they sort of illustrate like with arrows, the genes and the directionality of those genes. And then in the layer below, that's the proteins that they make, right? So they make these two ESX proteins that they have that motif WXG100. It makes the toxin CPNT, which both has the channel part and the toxin part, and then the antitoxin. And so to study it, like a good genetics lab slash biochemistry, they're going to make a knockout
yeah,
and remove the whole operon from their Mycobacterium strain. Um, and uh, they remove it. Oh, actually, I do still want to just talk
all right,
about
hold on.
A and B there. They remove it and they want to show that it's removed. So that's what A is. It's showing that they removed it with PCR.
Yep.
Normally, you'd want to see like a big chunk of DNA there, but they remove it. So now there's a small chunk. And then they do like I think this is a sanity check. I don't know
Yeah.
if you read deeper into
So
this. The B.
PDIM, which stands for phthiocerol dimycoseraceates, which are key components of the
Okay.
Mycobacterium outer membrane. So I think this is a sanity
Yeah.
check to make sure that when they knock these out, they don't affect the rest of the cell. Because we know
Mm-hmm.
that the CPNT has multiple functions, so it works
Yep.
as a normal channel. So if it causes another effect that causes the cell to become sick, then the researchers need to know that. Otherwise, they or the rest of the experiments they do might be kind of thrown out of whack. So making
Yeah.
sure that that they're isolating this system essentially, that they're
Yeah.
looking at just this and what the effects of these deletions will be happening. So yeah, um,
Totally. Yep. Yeah. So they successfully made this knockout, and then now we can, I think, move on to the next slide. To then understand that knockout, they want to put back in each component to see what does it do when you put them in isolation. So they put them on plasmids, little mobile pieces of DNA that just have either ESXF by itself, ESXE, or the toxin antitoxin pair.
yeah, and so so they find that ESXF and E seem to only be available in the whole cell. This is whole cell lysis when they're both there,
Mm-hmm.
and
Yep.
CPN is also there kind of the whole time, although it's only there at higher levels when these are there. Uh,
Yeah. I think it's interesting that they're dependent on each other, right? That you can't
right.
have F without E. So yeah, they probably like are somehow like interrelated in terms of stability. Like if they don't come together, then one just gets degraded, is my guess. But there could
Yeah,
be many things going on.
and also they kind of CPNT. As we know, it messes with NAD plus. So if you put
Hmm.
NAD plus in like kind of medium with this, then
Yep.
if it gets destroyed, we know that we've got this tuberculosis necrotizing toxin in there, and
Yes.
they do heat treatment. So heat treatment is usually there to kind of unravel enzymes,
Hmm.
and so we can see whether something is active or not in that by saying, okay, well, we've got this CPNE, and if we heat treat it, then it no longer breaks down NAD. Um, uh,
Yeah, I think actually they're seeing. I think the idea is here that with the heat treatment, the delta. Oh yeah, no, there's still a little bit of activity, right? Like the heat
yeah,
treatment, I think removes the activity of IFT, is what they're saying.
yeah,
Yeah, and the CPNT still resolves. They're trying to understand in this figure. I think they're trying to understand like why don't they see a lot of CPNT on the graph? Oh, right. Why like is CPNT dependent on making ESXE and ESXF?
right,
And in this, they're saying like, oh, maybe what's happening is something's going on with its interaction with IFT, right? Like when the IFT is around, um, it doesn't it doesn't function as well. It doesn't show up on the Western.
yeah, and also they've got this deletion of it where they since they delete the ESX, suddenly the
Mm-hmm.
CPNT no longer works. Tuberculosis necrotizing toxin is no longer active, so that also
Mm-hmm.
kind of indicates that there is something going on there. Um,
Yeah, yeah. Not active, but still, yeah, not active.
yeah,
Yeah, I guess. Oh, so I guess yeah. Sorry, maybe I misspoke then. Let's just say that the graph, the C is really just about does it work, right?
yeah,
In what state does it work? Yeah.
okay.
Okay.
All right, so DEF. So this is where so first part of it I think is flow cytometry, where they
Mm-hmm.
through like this machine that laser reads like whether something's expressed or not.
Yep.
Um,
Yep. And probably on the outside, more or
yeah,
less, they're thinking about the outside of the cell because like they don't permeabilize the cells at all when they put them through this flow cytometry. So they only can bind when they're looking for these laser lights to light up. Like they can only bind on the outside.
yeah. So they've got these
So
fluorescent
D is actually.
antibodies that have to bind
Mhm.
to the outside of the cells, and since these aren't permeabilized, those can't go inside the cells. So can only detect stuff that's right on the surface.
Mm-hmm. And that's the same thing basically in E, right? Like D is the laser way of looking at it, and E is like the lasers, but then we use our eyeballs to take pictures,
Yeah,
and then just like count the puncta, like the sort of the red dots, right? Are the TNT domain.
yeah. So this is a microscopy of the Mycobacterium, so you can see that these lines are the Mycobacteria, and these
Mm-hmm.
puncta are just like concentrated areas of necrotizing toxin. Um,
Mm-hmm.
yeah,
Yeah, so it's like they're looking for is TNT on the outside of these cells, and it seems like that's the issue, I guess, like that they're seeing is that it can't get to the outside anymore when they remove ESXF and ESXE.
yeah. And in Figure F is basically where a bunch of researchers will look through like maybe a couple of images of maybe like could be hundreds of images of this, and then they note down their
Yeah.
results, and then they do an analysis of those. I think they counted
Mm-hmm.
like over 1,000 cells for each ones, and to try
Yep.
and figure out like whether there's like a general difference. And it seems
Yes.
like yeah, there is. Um,
Mm-hmm.
so you need to have like ESX, sorry, ESX, ESXF,
ESX,
ESXF,
F and E.
F and E
Yeah.
both in order to have this external TNT toxin.
Mm-hmm. Mm-hmm.
And now going to
Maybe this
this.
is more realistic scenario. So like they look at macrophages, which are the cells that eat tuberculosis, right, where TB wants to live. And so you would assume that the TNT on the outside is supposed to be for these macrophages, right? Like so they put them in this more realistic scenario and they do the same, they do immunofluorescence microscopy again. So they look at these cells, they did the little white dotted lines, tells you like the cell boundaries, and you can see the MTB is lighting up green and the TNT toxin again. Oop.
Oops.
Sorry, that's my phone.
It's all right. So this time they've actually permeabilized the macrophages, so the antibodies can go inside, and we can see the intracellular TNT. So we can see whether it's being produced, not necessarily whether it goes outside the cell.
Yeah, well, no, this is permeabilize the
The
macrophages, right?
macrophages,
But
sorry,
not
yes.
necessarily permeabilize the MTB.
Right, yes,
I
of
think that
course.
it's like hard to permeabilize MTB. I'm not sure if they're going to look at that. Like, I mean, that's not even, they already have information from grinding up things and looking at the Western blots. They don't have to permeabilize the MTB.
Yes, you're absolutely right. So yeah, this is inside they permeabilize the macrophages. So now we're looking at TB in its almost native environment, which is inside
Yeah.
of a macrophage.
Yep. And they see the same things, right? That when you don't have ESXE or ESXF, they don't make this T out into the macrophage cytoplasm.
Yeah, these are. I mean, it might seem to be a repeat of the previous experiment, but actually these are quite important because almost the first experiment is just to screen out whether if it doesn't have an effect in the previous experiment, then we need to know. Then we don't need to do this experiment.
Yes.
But once we have that result, we have to build on that and say, okay, well, move it into more and more realistic settings to try and figure out
Hmm.
how this bacteria is behaving in a real life situation.
Yes, absolutely. Yeah, this is the closest they get to real life is going to
Yeah,
be inside of THP1 macrophages. And I mean it's pretty good. It's
yeah,
not exactly, right? So like the caveat always like this is if we talk about infectious models, this is their infectious model, right? This is their mouse. Is a cell line, right? That TB infects. Yeah,
yeah.
and
So
then the little western blot below that is just in media. So it's also the same in media that they see the same effect. If you don't have ESXX or ESX, oh my God, it is hard to say
yeah.
ESXS E or F, then you don't get secretion of CPNT into the
Whereas if
soup.
you actually grind up the Mycobacterium and pop those cells, you can actually see that it is still being produced inside these cells. It's just not
Yeah,
being released into the wider media.
totally.
So
Okay.
that brings us to figure 2A, where we're going to focus in. So now we've kind of, with those big knockouts, you've established that ESXE and ESXX, the two proteins, we established that these are both important, but that's very, now we're going to narrow down even further to see what part of these proteins are important. So is it
Hmm.
the actual WXG motif that is an important part of how this protein functions the way it does? Or is there something else? So
Yeah.
ESXX, ESXF has a YXXXE motif, which is a secretion signal of the substrate. So the question is, what effect does that have?
Yes. Yeah. So they have three things that might be responsive, right? Basically, three hypotheses for them, right? Of stuff that they want to know. Does this affect, right, the ability of these two proteins to, mediate this secretion event that they've discovered? So yeah, WXG and then the secretion signal YXXDE. So Y is like, ooh, tyrosine,
Tyrosine, I
and
think that is.
yeah, and then X's or anything, and then D and E is I
So I
don't know.
think D is asparagus. No, no,
Oh,
wait.
aspartic acid.
Aspar.
Aspartic acid.
Yeah,
Yeah, and then E is I don't know glutamine. No, we said glutamine.
we've already. Yeah. So
Oh yeah, no,
follow us
it's glut.
from.
You can always look this up. I think this is
Yeah.
an important. I want to make this point because, well, I've been talking about mutations with some other people. I mean, we talk about mutations a lot with a spike protein, right? But
Right.
like scientists don't have to memorize this stuff. You can just look it up on the internet.
Yeah. Follow us for more inaccurate amino acid declarations. So W is William. G is George. Y is Yvette.
All right,
But
let's see
yeah,
what we
no,
got
yeah.
next.
I can confirm the tryptophan and glycine are the WXG one because I just posted up on Google. But apart from
Oh,
that,
nice.
that's as far as I get with my research at the moment. So yeah, going into SXE and SXF in Figure 2BC, I figured out a way I can save them without
Oh,
tripping over.
Essex. That's how we're gonna
Essex.
say it. Okay, I like it. Essex.
Yeah,
Let's do it.
Essex. Right. So this one is adding. So now they've added in these mutations to disrupt like these motifs, and we can see how
Hmm.
they play. Almost very similar to how we saw in the original experiments.
Yeah.
Um,
And in fact, they're really just repeating it for us, right? Like they're gonna look at whole cell lysate and they're gonna see does it have an effect on CPNT or yeah.
yeah, and the effects are actually much more subtle. So because
Hmm.
it's not so much whether something's on or off, it's like how well it works. In which case,
Hmm.
so you can see some of these bands are lighter, some are darker. That relates to how much of this protein is available in the sample. So to order
Yeah.
to actually quantify that, they basically did graphed how dark these bands are,
Hmm.
and you can see that on Figure C, where they
Yeah.
looked at how intense these bands are.
Yeah.
And
They normalize it. So the idea is that like right in this, there's probably a lot of detail. We will talk. basically it's going to be a whole other talk to try to describe the whole Western blot mentality, but
Mm.
like the idea is that there's a control, the RNAP, like the darkness of that band, right, is more or less how much protein you've put in. And so like if all the darknesses you relate that, right, as a ratio of the darkness of the control band, then that's how you get these normalized intensities.
Yeah. So like you can see, like say in one band that's slightly lighter, and all the other none of the subsequent bands are going to be darker than that. If you
Yeah.
or they're relatively not going to fluctuate too much from that area, and that's how you kind of normal. That's your control. That's so RNAP is your zero, and everything up, every level of darkness is related to that. Um,
Yes. Yes. Yeah, so same effects that we're seeing, they're not like as dramatic as the full knockout, but it is true that with those mutations in ESX, with the WXG mutations rather, right? That's in the WXG mutations, they see a little decrease in the amount of CPNT. But in the YXXE motif, they don't really see as much of a decrease. So that one doesn't seem to be influencing this as much.
yeah, that's right. Uh, and so now moving on to Figure 2D, where we again take these mutants and look at how they work in a real situation, where they're
Mm-hmm.
inside these THP1 macrophages. Uh, so I guess I should put some backstory there. This is still a cell culture model, so these aren't from. They were from someone who had, I think, a
Some
leukemia
sort of
or some kind of
lymphoma.
cancer.
Yeah.
Yeah,
Mm-hmm.
lymphoma.
That's what I'm guessing.
But yeah, but they're a standardized line, so you can grow them. So it's what we can work with rather than what's the best model sometimes.
Yeah.
And with this one, we see some. Uh,
Well, Foz, I just want to say, I want to just lob in there. Like, I think you did a good job explaining that, you do you do sort of small experiments and then you build up the complexity to see if it relates to the real life. And so, right, like, yes, there has to be follow-ups from these sorts of ideas, but like, this is part of the building up process. And THP1, it's a great midpoint in some ways,
hmm,
right? A great way to show a little bit more relevance here, but then also bridge that gap. The gap will have to be bridged when we
yeah.
talk about real infection.
So yeah, again, we're looking at the levels of TNT in these macrophages, and
Mm-hmm.
we're seeing that.
Yeah, I think it's the same pattern, right? That with with the YXXG or the YXXE motif mutation, that's the tall, right? That's the second bar from the right.
Yes.
Second bar from the right at E. So like that one doesn't have a big effect. That's the same as wild type and the same as full complementation. And then in the other mutants you get less.
Yeah, so I think the takeaway is that there is something happening with the WXG, but with the YXXD, there isn't necessarily something that we're seeing.
Yeah.
Um,
Mm-hmm.
and that brings us to surface expression.
Yeah, they're going to look at surface expression as well, and um, they're seeing the same things.
Yeah, more or less. Uh,
So it's great. I mean, like this is the I think this is a good illustration of how science sort of gets done, right? You
yeah,
sort of choose the set of tools and the things you're going to do, and then you ask a question, and then use those tools to verify.
yeah. I mean, theoretically, all these three kind of images should look the same because they're using the same proteins. Essentially, they're all being
Mhm.
expressed, but just one or two mutations are suddenly in the ESX. I mean, they haven't done both of them. They've done one at a time, and each of them with this WXG motif always is very important for what their joint function is.
Yes, yes.
Yes. And,
I guess. Sorry, I just want to say at this point, uh, it could be joint function, or it could be, yeah, like I guess it is. We know that it's joint function because the removal of one impacts the other one. Even though there's a functional version of the other one, it still doesn't
hmm.
rescue anything, right? Yeah. So there's that idea that are necessary. They're interdependent.
And that brings us to 3A, where we're looking at the soluble and membrane fractions of the Mycobacterium. So
Yeah.
most like kind of bacteria are formed of like two things: the membrane, which keeps everything like kind of inside,
Mm-hmm.
and then the insides of the cell, which are generally soluble, and where all
Yeah.
the interesting chemistry happens. Well,
The
some of it,
the
but now we're
cytoplasm.
learning,
The
yeah,
cytoplasm.
cytoplasm. Um, and now we're learning more, lots of interesting stuff is happening in the membranes, and so trying to figure out where is ESX and ESSX, so SXE and SXF, where are they going to be doing their jobs?
Yeah. And I think you can probably tell, like, by this point, right? Like, you can sort of, I mean, I think the authors are saying it in their paper too, right? Like, they think that ESXF and SXF and SXE, like, they are helping in secretion. And so if they're helping in
Hmm.
secretion, they should be in the membrane, right? Like, and so knowing where they are in these cases, like, is helping to build that story of like what exactly are they doing. We want to know where they are so we know what they're doing.
Yeah, and they've got these controls to basically prove where they are, and so like there's one that's only soluble in the like kind of soluble fraction that's in, and the other one that's generally oh, that's CPNT, sorry.
Yeah,
Mixed B is the one that's only in the membrane.
that's right.
CPNT again, seeing where that's being produced in these different strains,
Mm-hmm.
uh, and where it lives, which is
Yeah.
interesting.
And
So
it
the
is
wild
and it
type,
is in the membrane and in the soluble. And that makes sense, right?
yeah,
Because we know that it's a channel protein that has like a little soluble piece that falls off. So
yes. That brings to
oh
figure
wait,
3b, where
I guess we do want to say what the conclusion they drew from that last figure is. It's just that so they have their mutants again, right? That they tested through this thing, and looks like the mutation actually doesn't change the amount that's in the membrane or that's soluble. Not appreciably, at least. Maybe like a little bit less in the membrane. It's hard to say. I feel
yeah,
like they didn't give us the densitometry, like the normalized stuff, so like it's not easy to say from this one.
indeed. Figure 3b is where they use a chemical crosslinker. So when proteins bind to each other, having that chemical crosslinker means that they stay together. So when you pull them out of the
Mm-hmm.
solution, you can see exactly what kind of conformations they have.
Yep.
Uh, so
Uh, and in this one, when they use the crosslinker, they don't get a lot of these ESXE monomers, look a lot, they're quite low, right, in
right,
those three senses. So that's a little clue that like ESXE is like getting bound up into bigger groups in some ways, right?
right, and like so when you have these like kind of without the cross-linking, they're basically there's one size of ESXE, so they all group in the same way. But when you have oligomers, they can link up in chains, and so they can be any size. So you get more smearing of all these. You get much more diversity in the level of sizes, so they don't all bunch together in one area.
Yeah, I mean, we don't know what size, what shape they would be necessarily,
No,
right? Like just that they clump, they're
yeah,
clumped in some way. But it's interesting that they point out that the CPT is also like trapped in this complex as well, right? Like when they add this crosslinker, CPT is in this mix, right? Which I guess
mm,
makes sense. It should be interacting. If they have some effect, that they have some sort of physical interaction in the cell. And I guess importantly that those mutations do disrupt that a little bit, right? Like
yeah,
that dark smear of CPNT, it's not really present in the two
yeah.
mutant columns.
And now, disrupting these motifs doesn't significantly change the surface accessibility of SXF.
Yeah. So it kind of does, right? Because those bars are a bit lower. If you look at into so the gray bars, right?
Yeah,
SXF. Like the gray bars are kind of going down, but they're not significant. They're all significantly different from not having anything. They're all being compared to the case where there's no surface expression.
right. Yeah. So again, this is one of those things where we could go into the weeds and say, okay, the statistical test here wasn't really designed in the way to
Yeah.
make a call one way or the other on this. So
Yeah.
this does leave things a little bit up in the air. Um,
It does. But I think like to me, this is like I think this happens a lot in science.
hmm,
Statistics kind of shitty, but they do it in multiple ways that gives you that confidence that okay, they've seen it from different angles though, so I'm buying the story.
yeah, I
Yeah.
I don't really see too. So I can sort of see where they're coming from here. There is some subtlety in there, but again, we didn't do the experiments. We don't know how many times this experiment failed, so we don't know.
Uh-huh.
Like maybe they've got more idea of saying like, okay, it looks like this is we've given this because it's our best image, but in most of our experiments, it didn't work. So
Yeah.
I mean, we can never really
And so
speculate on that aspect of it.
in the ESXE though, the pattern looks a little bit different.
Yes.
In that they don't really find a statistical difference. Uh, in or sorry that difference between no surface expression and what the expression that they get when they make where were the mutations? It's hard to read for me.
So this was the SXF
Hmm.
mutation. So no, hang on, No, wait. So this was
ESXE.
SXE.
Yeah, but the
So
mutation that they make is in F. Yeah, the mutation they make
yeah.
is in F. So when they make the
So
mutation in F, that seems to somehow disrupt the ability for E to get surface localized.
right, yeah, I
Yeah.
think so. I'm because this
Hmm.
is one of those things where there's so many things to keep it in up in my head that I could because so like in ESXF they've disrupted like well so ESX SXF they've disrupted here, and because of that suddenly you've got
No, so
these
those are
significant
the
difference.
antibodies, right?
Oh right,
Right. So what we're seeing is the level that they see it at. So in the gray, they're seeing F, right? Like the bars are high because there is F.
right,
And then in E, the bars are low because there's no E. But if you have to
yeah,
then read the rows, which are the really hard things to read. But if you look at the third row from the right, right, that's a low row and that has the mutation plus ESXF W whatever. So that's a mutation in F that affects E. But if you look at the row to sorry, the column to the rightmost column, that one has the mutation in E. Sorry, I'm looking at D here.
oh sorry D, okay,
Yeah. Right. I mean, it's correlated, right? It's correlated.
yeah,
um,
okay,
yeah. If you look at that row, that one shows that it's the mutation in E. The WX motif in E is removed. Yeah, and that one doesn't seem to have as big of an effect. Right. I mean, we're sort of building this interdependent story, right? There's nuance
yeah,
in it, right? Like, yeah, if you make the mutation, it messes things up. But actually, there's when you mess it up in this way, it actually only affects E for some reason. So yeah, there's don't know what it
interesting.
means yet. It's just describing this particular conundrum.
Right, so 3EFGH. So this is going back into to the macrophages, looking at
Hmm.
the difference, the reduction in SXF secretion,
Yes,
um,
and
because
SXE
yeah,
as well.
yeah,
Yeah, except again, now we're in macrophages. This is the better system, presumably, and I think they see the same thing.
yeah.
Yeah.
So kind of the takeaway they get from this is that these two proteins are interdependent, and maybe they're forming some kind of complex that and that's what's needed for it to be expressed on the surface.
Hmm. Hmm.
And that's dependent on the WXG motif partially.
Yeah, and I guess like for some reason the WXG motif in the ESXF is like quite important for ESXE.
Yeah,
Again, this is about interdependent, right? We know they're interdependent, but like how are they interdependent? So these are kind of these little nitty-gritty biochemical details, I guess, born out in this it's a genetics approach
mm,
to try to understand what's going on.
yeah, and that brings us to figure S3, where they start trying to model this interaction. I think,
Yeah.
yeah,
So they have structures of an ESX EF pair in a different mycobacterium,
mm,
Mycobacterium abscessus. And so they can use those structures to basically guess at the structure that ESX E and F in tuberculosis will form. And it's this structure of these helices, which is very common for things that insert into membranes. And in fact, when they do these little prediction algorithms, they find that there are these transmembrane predictions. There are predictions that say that certain parts of these helices should be in the membrane, and
mm,
also some of these helices are of a sort that associate together, so they clump together and they try to stay away from the membrane, right? So that's the idea of something that's an intermembrane or something that's a transmembrane protein will have these sort of characteristics. They'll get clumped
right,
up. Certain parts will want to clump, and certain parts will have to be exposed to the membrane that they're inserting into.
right. That brings us to some more gels looking at
Yes.
SXF in like,
So I think it's good to just pause now and say they've built this story about the genetics, right? Like when they mess up these different things, like they see a change in the phenotype, right, of MTB. But what they're going to do now is they're going to say, okay, well, we want to know more precisely what is going on with ESXF and ESXE, because we have these predictions that say they have these features, they're going to get inserted in membranes, and like we have this idea that it's going to be, you know, something that gets into the membrane to secrete stuff. So how do we show that? Well, they're going to go through the biochemistry route by making a bunch of this protein and testing it in different scenarios. So that's what we're doing here in 4A and S4AB. They're making the protein.
Right. So this basically shows that they're making it. Well, they're
Yeah,
making. Yeah.
they're making it, and they're pure. The middle panel is the purification scheme. So like you can see that like they have uninduced means that they didn't add anything into it. Then they have induced means they add the inducer. Proteins getting made. The bands are all darker.
Mm-hmm.
Then they use this amylose resin. That's because the ESX, F, and E are tagged with something very specific that just binds to amylose. So all the proteins now binding, and they get flow through, or sorry, they get like a little bit of stuff. They use TEV to cleave off. This is like
Mm.
basic, biotech sort of things where you like then cleave that protein off the column. Yeah, then they put it through some different flow columns basically to get it even more pure. And at the end, you can see that they get this increasing amount of ESX, F, and E by the end. Those two bands.
Right. Yeah.
Oh
Uh,
yeah, MBP. That's what's binding to amylose, uh, right? Is the maltose binding protein. That's part of
right.
their purification strategy.
Yeah. I mean, this is something that is quite new to me. I never really did much of this during my PhD. So
Oh
all this.
really? I love
Yeah.
this stuff. This is like I feel like this is what brought me to infectious disease. I actually started in a crystallography lab,
Oh
so like
wow.
we just made proteins all the time.
See, I was the opposite. I was mostly like a kind of a pathogenesis person, and I did like this once where I had to like do this all in the fridge because I think the apparatus only worked properly when it was cooled down. So having these protein experiments whilst in this cold room where it was like really cold and having to like elute protein through multiple fractions.
Yes, that's
I
right.
yeah I after that
So
I
you've
did.
done so you've done a process like this, but it
Uh,
wasn't a big part of your work.
it wasn't a big part of my work. It was a very small part of my work,
Uh-huh.
and that's partly because I didn't really like being in the refrigerator room. Um,
Okay, let's look at what we got next.
yeah. Okay. So
So when you run these protein gels, right, they usually have SDS in them, and that denatures all the proteins, so
mm,
they're all like floppy noodles going through the gel. But you can actually run these gels without the SDS, and that keeps it more in a native conformation. So the idea is that proteins have to be shapes to do their
mm,
functions. If you're adding SDS, it breaks apart the shapes. But if you want to kind of get a sense of what those shapes or those oligomers, let's say, are going to be, you have to remove the SDS and do this sort of test. So here they remove it and they show that, oh look, there's all these oligomers that form in the non-denaturing form. That's what they're seeing in B. In C, you can see that they're doing a gel filtration. So it's kind of it's a column instead of a gel. And they're running their proteins through and they see, oh, when they run it through, they get this high molecular weight thing. But then when they add GUHCl, right, which is a denaturing agent, that
right.
would break apart all that high molecular weight stuff and they get a smaller weight. Okay, so this is another piece of evidence to say, okay, it's clumping.
Yeah.
The protein is clumping. It wants to stay in groups.
Right. I
um,
see.
and then so what we have in G and H, I guess, is just that idea that like a fresh lysate gives us this high molecular weight. But if you wait a long time, like 4 degrees Celsius for days, 2 days
Yeah.
in that refrigerator, don't worry, you don't have to stay with this protein. It'll just sit there by itself. You get a lot of these clumps. So like there's a tendency for this protein to want to clump up, I guess is what they're getting at.
Right. Okay. And so differential scanning fluorimetry, which is, I guess, where they add they so proteins generally fluoresce mostly because of so I think one of them so some proteins I think the one they go for is tryptophan or tyrosine, which
Mm-hmm.
have these rings that fluoresce, and
Yep.
so and the level of fluorescence depends on the structure of the protein. So I guess well this works by looking at how that fluorescence kind of changes at
Hmm.
different temperatures.
Hmm. Yep. And the more things get denatured, the more those things will be exposed. And so what they can get a sense of is, oh, these proteins are folded in some way, right? So
Hmm.
like as they increase the heat, as they add more denaturing agent, they get more fluorescence, right? So they know that what they are purifying from the beginning point, from the cold, right, is something structured. It's not just like a disordered blob.
Right.
They're making it disordered, and that's giving them a sense that they have something that's well folded. Mhm.
Mm.
It's like a quality control, I guess.
Yeah. And
Yeah.
now we're going to the ultimate build-up of that, which is looking at it
Yes.
under transmission electron microscopy and
Yeah.
looking at seeing that actually these clumps that we've been seeing on the gel, what's forming are these kind of these five component like proteins, I guess.
Yeah, some sort of ring formation with 5 units and a little channel. They even measure the size of the channel. So we've seen this before in cryo-EM. This is actually not cryo-EM. This is straight up regular EM.
Mm.
So, this stuff gets embedded into like a resin and gets dyed with heavy metals. But all that's pretty small in comparison to a protein. So you get to actually see proteins in all of this.
Mm.
And you can take many pictures and average them all together to get these class averaged images, and then from that you can kind of get understanding what they look like. So yeah, there's some sort of pore thing, but they also notice all these strands. So there's other stuff too. So like their interpretation is the strands are probably the highest molecular weight stuff, and the pores are probably like some middling molecular weight thing. Yeah.
Yeah. So because they might form pores in the membranes, they decide to do these lipid bilayer experiments where they kind of have an artificial like kind of lipid like kind of layer that they put the pores
Mm-hmm.
in, and then they test whether that can because lipids are generally they're insulators, so they don't carry charge very
Yes,
well. But if you
that's
like
right.
poke a hole with them using a pore, they can carry charge. So by introducing these oligomers, they can test whether they can form these pores in the membrane.
Yeah, totally. And it's like a really interesting technique because you can see that as pores get inserted and like open up, there's a discrete jump of current transmitted. And so you actually get a lot of data from this measuring technique. You can sort of say, oh, look, pores are getting inserted or pores are like opening, right? Like
Right.
increase in current is basically the allowing of water or something, some sort of ion to pass through.
Yeah. So yeah, and they decide to test that even. So this is like some more
Oh
background to
yeah.
look at because the buffer matters a lot in these sorts of
Mm-hmm.
experiments. Because
Mm-hmm.
again, you want to have the right solution for the pores to act because we don't know what's comfortable for those pores, I guess.
Yeah, so they find the best technique as this phosphate buffer. Simple. Let's move on.
Yeah, yeah. Move. And looking at pH dependence, so changing the pH of the medium to see whether that affects how often these kinds of little pores open or close. Um,
Yeah. So I this is interesting to me because like there's kind of two reasons to do this. One is protein structure is dependent on pH, right?
mhm.
So like of course like pH probably matters, but the other one is, living inside of these liposomes, right? Like when you get engulfed for tuberculosis, like the fate of that is supposed to be acidification and death.
Right.
And so like part of living in that environment is sensing pH. And so like I also think that's a really interesting angle, right? That adding this data adds into it, right? We're also understanding maybe something about what might be the response inside of a scenario it could very well encounter.
Yeah, it seems like as the pHs get lower, the
Hmm.
pores open or close more often. They don't open up; they just close up. Whereas at
Yeah,
higher
they don't get
pHs,
inserted as much. Yeah, but
so
but there's a sweet spot, right? Like at this,
yeah,
if you're going 7 and up, it's not even right. It's not working there either. I don't know why they don't go more to the other side. Anyways, let's.
I assume they found their sweet spot, and either side they just didn't expect that. Say if we make it more acidic, is there going to be a second sweet spot? I feel like
Yeah,
maybe they didn't
I
want
think
to,
it's probably physiologic. There's probably no time where it ever would go up to 7 inside of this compartment.
or even like go down to three. I mean, because I guess the thing we'd be worried about if I'm a TB inside these cells, we want to avoid the kind of the acidification. So you
Yes,
want to release it at a
they do
point where
avoid
it's
acidification.
yeah.
Yeah, they do avoid it.
Uh, and also, so I think this is an important factor because, as we mentioned before, pH can affect the protein structure. So are these proteins just like getting messed up, or is it an
Hmm.
actual like kind of deliberate opening and closing?
Yeah. Well, actually, it's not an opening and closing, right? Like
Right.
that's the other thing that they show here. That like it's not about the. So what they do is that in pH 6, they allow all those. They allow it to get saturated, right, with as much current as it's gonna allow to flow. Then they add the pH thing that lowers the pH, and they say if they add it, then does it mess everything up, right? And does the current go down? But it doesn't. So
Mm,
that means it's not gating, right? It's not that the pores are opening and closing. It's rather the pore getting in to the membrane. That's pH dependent.
right.
Yeah.
So yeah,
Yeah.
yeah. Oh
This is
yeah,
actually, I
that's.
think, this is a really important control in my mind because
Mm,
this is a part of the field that they're speaking in. Actually, they're talking about pores, and like there's a lot of mechanisms. So like the lab that I came from is an alpha hemolysin lab. That's like a big, well-studied pore, and there's all these different things people know about how they work, right? And so pH dependence in terms of opening and closing it, that's one of the things.
right. I see.
Yeah.
Yeah. That's quite cool. Next, they make artificial liposomes. So
Yeah.
taking that membrane before and just turning it into these blobs and seeing what happens when you add these proteins to them.
Yep. And they look at it under EM first and they say, okay, they can see these pores. They're getting inserted into liposomes, and the liposomes get all messed up. All the blobs don't look as perfectly circular anymore. So that's the difference between H and I. And then they start zooming in. In K, you can see some zoom ins of like a pore. In M, they're saying that's a cluster of pores. And in N, they're saying like, oh, look at they even see some of those long filaments maybe attaching to the surface. But these are all just
Yeah,
pictures.
I think that that's kind of an interesting because like with N, I'd be tempted to say, oh, that's just an experimental thing, rather.
Mm-hmm.
So I mean, again, this is still not necessarily a natural system, but it is
Mm-hmm.
interesting that we're seeing it in the as we get further closer to what a real system looks like.
Yeah, yeah. It's like they're just characterizing. This is a lot of this is like the biochemical follow-up, right? This is how I see the paper. We're in the biochemical follow-up to the genetics that they did.
And figure 4EF, so giant unilaminar vesicles.
It's such a funny term. I didn't even know that these things existed, but I guess they're bigger than liposomes, so you can look at. This is a light microscope now. They didn't have to do TEM to see them, and so like even on this large scale, they see the same stuff. When you add ESX E and F, SX E and F to it, it breaks the big bubbles.
Yeah. So that means
Hmm.
so stuff is coming in and out, which is
Yep.
disrupting the structure of these bubbles. So you're getting
Hmm.
them looking very odd conformations.
Hmm.
And so that was and now we're going back to the
Yeah,
SXE and SXF.
I mean, this is it's all being SXX and ENF.
Yeah.
They, so they look closer at the structure, right? To because really they're building. They know all of this stuff is done in light of knowing all this
Hmm.
thing about pore forming stuff, right? Like they are determined to show that ESX, E, and F are making these pores and these pores have function, right? And one of the ways that they could have this function is the hydrophobic tip. So they see like they categorize all the amino acids by how water loving, right, or how fat loving they are. And
Hmm.
in lipid bilayers, there's fat, right?
Hmm.
There's fat in the middle. So in order to get in there, you have to be able to have some hydrophobicity. And so they say, oh, look, like let's focus in on this section, this hydrophobic tip. Maybe this is how the complex gets in. And what's interesting is there's also a GXW motif in
Hmm.
ESXE that's at that tip. So they're like, oh, maybe we also missed something. They're
Hmm.
kind of like, now that they have a better understanding of how it's going in, they're like, well, we also want to make that mutation to see if that's affecting how things work.
Right.
So they're really, I guess, from that they already have like a very strong hypothesis that like there's going to be an insertion event right at the tip, and they're going to break it to see if that's the case. And just this figure here is what's really interesting is that GXW motif is conserved amongst all these similar to ESXE proteins. And these are paralogs, so this is
Hmm.
not between organisms. This is all in MTB. So MTB has duplicated, right? As I said, there's all these different ESX systems that got duplicated. In all of the systems that have an ESXE protein, it has these two conserved domains. And so again, if we think back to the introduction, like what don't we know about tuberculosis secretion, right? There's this mystery that's going on, and they're excited that they may be saying something to that mystery, right? And that it could be applicable to all the ESX systems that are studied inside of this organism.
Indeed. And so they did mutations again.
Yep, and purification. So
Yep.
they repeated the stuff. They made the mutants, they purified the proteins. You can see on the anion exchange column, you can see on the gel.
Yep. And then they looked at the actual insertions. Well, the events using again that lipid bilayer model.
Yep. And the mutation of those two domains does remove the ability for these proteins to insert.
Yeah.
But an interesting observation, which isn't very big on this screen, but I wrote it down so I would remember to talk about it. If you look at figure D there, or sorry, on the right-hand side, D at the bottom, they actually, I guess, I don't really know how they can separate insertions from openings and closings. I think it has to do with like the fluctuation of the current that like, I guess current like has a frequency as well. It has something to do with frequency. Anyways, but they see that there's a lot of openings and closings, right? There's still a lot of numbers. There's like 200 or something in the later ones. So while the insertions aren't high, they still have this high number of openings and closings, which sort of suggests that it's still getting into the ones that get in the membrane are doing something, but they're just not doing it right.
Yeah. This is so the question is where the barrier is. Does a change the structure mean that it can't insert into the membrane, or once it's in the membrane, can it do its job or not? And it's a bit of both by the looks of it.
A bit of both. In that mutant form for sure.
Yeah.
Yeah,
So
and in the other mutants, it doesn't even get in. It seems.
Yeah.
Or that's a really high. Yeah, getting in is the big barrier.
But I think
Sorry, the big barrier is getting in all the mutants, but in the last mutant that they describe, there's also something where it's partially active.
Right.
And again, I want to relate this back to like there's a lot of pore-forming literature, but like studying the behavior of these mutants is a great way to understand how the pore even works, right? These are very important tools to go forward with this type of research. Okay.
Okay, so looking into like liposomes once more to see whether they deform them, and these mutant oligomers, they don't deform the liposomes, although they do seem to be in there. So we can see on this electron microscopy, they point to areas where they kind of seem to show that these pores are in there.
Yep. Yeah. So the pores seem like they're there, or something's in there. This is like zoomed out. I wish they zoomed in for us so we could actually see the pores. They will later, but the liposomes are still happy little balls. They're not the same like popped like mess that happens when you have wild type added. So.
Yeah, and the complex seem to. I mean, I think they note that the complex seems to be slightly different in these versions, where like
Yes, and
yeah.
I think they'll show it in a little bit later, but they didn't show it in this figure.
And again, the giant unilamellar vesicles. I think that's what it was called.
Yep.
Again, they aren't affected. I mean, wild type like causes them to like disrupt completely, but the rest of them, I mean, ESA. I mean, again, the one with like the single mutant that has some partial activity, we see something that looks like it could be disrupting.
Yeah.
So
I think they just chose that picture to like give us a teaser because, because like the graphs like I don't know, it's like it's hard to say from single pictures. They're definitely teasing something there.
Yeah, the graphs don't really show the same thing.
Yeah. They show us one picture and they're like, "Oh, does that mean there's a different?" It's like some weird subtle reinforcement, but I'm like, I'm not believing it. Show me some different data.
Yeah. Show me different data. Which is looking from figure SCEDF. So this is looking at quite a lot of TM images of the mutant and wild type versions of SXF, SXE.
And this is importantly that those class averages, right? So like after seeing so many pictures of them, right, they kind of come to these consensus images. The things are looking
Right.
different. I guess that's
Yeah.
important.
Yeah, you can see with the mutants, like kind of with kind of the normal one, you can see this very kind of stable, like kind of conformation fairly. Whereas the other
I mean,
ones, there's
they're all
actually
stable.
yeah, looking at
Like they should be the pictures that they show. They should be all stable conformations. Otherwise, they wouldn't be able to make a good class average, right? Like
yeah,
so,
that's
like I
true.
mean, I guess you can see in D and F, like there are some pictures that don't look like pores at all. I think that is a good indication that like there's something wrong there because if you can't even make an image, it means that there's just too much noise, right, to have a good average thing. But if you do end up seeing a particle by this method, then something is being made.
Yeah.
Yeah. And I think I just realized that's probably figure S7, not full figure. There's no full figure 7. Anyways, not that it
Right.
matters.
Yeah. Sorry. Yeah.
No, that's me. I made the slides. Um, and so in this, so with those class average pictures, they can reconstruct a structure. So this is exactly how cryo-EM gets us really good structures, but they're not even using, they're using regular TEM, and
Mm.
their pictures are not even, they don't collect enough to get a really high resolution thing. So what they end up creating is something that's probably a little bit lower resolution than you would get in cryo-EM structure determination. But it's the same idea, the same technique, right? The fact that
Mm.
every image is seeing this molecule from a different angle, it allows you to recompile those views and give you the three-dimensional structure. And then they manually go in and they say, okay, so we have our predicted model of the heterodimer of ESXF and ESXE. Let's fit it into the electron density map, and that's like an approximation of what we think what that pentamer might look like.
Right.
Mm-hmm. So it's a
And
very
then the
puzzle. It's a puzzly thing. They did a puzzle,
yeah.
but that's
So
structure determination for you, right? I don't know
mm.
how at length we've talked about this, but you know, I was in a crystallography lab before. That is structure determination. It's all about puzzling out from the data what something is.
So yeah, I'm just
There's always
imagining you like kind of with various structures, it's like trying to link them together in different ways to find the right Lego conformation to get it to form into a stable shape.
Yeah, that's how they do it with computers, right? So it's
Mm.
not physically. So here they did it manually, so they actually did probably did some some dragging, but like when they do, yeah, when they do that determination, computers do all that. You know, they rotate everything, they spin things around, you put different constraints on it. So, anyways, the assembled structure that they get is this 17 angstroms. You know, that's like good enough for these purposes.
Yeah.
Pentamer structure, and you can see how the ESXF and ESXE sit in there, and how the different how there's an interface, right, that has to form between those oligomers.
Right.
But again, none of this is so this is but you have to really understand where the structure is coming from, right? This is not the definitive structure of such an object,
Mm.
right? Because what they've done is they've manually slotted in what they think is where these predicted structures would fit into this particular electron density. And then also remember, they don't like these are not in real cells. Like they've reconstituted this from beginning, and so we don't know like if this is the thing that gets made inside of our bodies, right? They didn't see this in the THP1, for example, right? They didn't do thin sections of THP1 until they found this picture. This is all from the biochemical stuff.
Right. Yeah. Moving on. They did do some Rosetta data to try and get it to the lowest energy level to make it like
Yeah, this is a type of confirmation, right? They actually
mm.
de novo. They said, okay, so let's assume they told Rosetta, let's assume it's a 5-member object,
Mm.
right? That has to come together with these predicted structures of ESX, E, and F. Show me what that would look like.
Right.
And then Rosetta starts playing around with the energy levels and says, okay, this seems the lowest energy way it could come together. And lo and behold, it looks like what they took a picture of. So they feel good about that, right? But again,
Yeah.
it's just it's another way of getting at it, but it's also a very in silico based approach.
Yeah. Uh, and then moving on to again differential scanning thermometry again to show the structural stability of this sample to uh,
Yeah, they lose me a little bit here because I don't think that these figures this is not the classical way you would show salvation. right? Like there
Right.
are very good biochemical techniques to tell us what is exposed to solvent and what isn't. They're using the tools that they have at their disposal to begin to tell this story, but this is not the way I think that the literature would generally accept this story to be told. And
Right. What?
so, yeah,
Yeah.
again, they're looking at whether or not they get denatured through temperature, right? But even just the baseline at regular 35 degrees, that they're saying that that absorbance is also telling you how open or closed the protein is, right? Which
Hmm.
is true. It's not incorrect. It's just like I'm not sure if people do it like that in terms of like a gold standard.
Right.
But what they see is that as they increase the heat, right, the mutants do end up deforming, but they deform in different ways, right? Like
Hmm.
some of them deform at an earlier temperature phase, right, and some of them deform maybe at a later temperature phase, and that could indicate more or less stability in the oligomer structure. Um, so again, they're built. It's a good way to start off with
Hmm.
this line of thought. But the idea that they're trying to say is that there's a sweet spot, right? That the biochemical characteristics of this oligomer, if you mess it up by making these mutations, you change those characteristics, and then ultimately are unable to make the pentameric form.
Yeah, I think it does just say a bit about the stability of it. So, and how
Mm-hmm.
much a certain mutation makes it easier for the protein to pop apart at earlier temperatures. And it
Yes.
definitely does say a lot about the SXE mutation being really weakening it and the W89A point.
Yep. Mm-hmm. Mm-hmm.
And maybe less so to the ESXF. But yeah, in terms of like the solvation, I'm not sure whether like because that's what baffled me because I'm not sure how that related. This mostly tends to be very looks like looking at how stable the structure is.
Yeah, yeah. I don't like that they use this particular wording. I would have liked that
Mm.
to be like restructured a little bit, um, or at least speak to the fact that like there's limitations in all the ways that they use. When people do this in the bio, I think we've seen how this is done. We've seen it in the paper that we've read previously that's most related to this work is the H2 the ACE2 decoys,
Right.
the soluble ACE2
Yeah.
decoys. They do a lot of biochemical characterization there, and oh, but that one doesn't have it. I think in an earlier one we looked at antibodies binding to to the spike protein and looking at whether or not those interfaces were open or closed. They
Mm.
did a real salvation like test there where they use like I think they use tritium, right? Like you
Mm.
soak the thing in something radioactive and then you use NMR to find out like where did that radioactive stuff get to, and then that gives you an idea of what's open and what's closed, where the surfaces are. But yeah, that's not what's happening here. They can't do it. Maybe they can't do it with such a large molecule. It could be too big. Um, but yes.
Mm.
Anyways, that's all. That's all I have to say about this subject. I'm not a big expert in it.
Yeah. And again, this graph is where I completely I gave up on this one because I'm not sure exactly what this is meant. What these comparisons are meant to say. I
So
sort of get.
salvation is on the x-axis, and on the
Mhm.
y-axis they have the frequency graph from the channel conductance, right?
Right.
What's going through. And again, they just want to show that there's a sweet spot, right? I don't think that it's also like in some ways it's like very obvious, right? Like if you break something, it will be broken. You can break it in different ways by making it more one way or more another way. In either case, it's broken, right? And it won't be able to work. And that's basically what this graph shows, right? That like unless the absorbance of the protein is sitting in this band, right? It doesn't make good channels.
Yeah, and they've highlighted like this area, this a middle area of where the situation is supposed to be similar to be in vivo. Like I think that's right. Or
Yeah, I mean, I
yeah,
don't know why they would say that is a in vivo section. They just like that's the wild type, right? Like the data that they're showing here is from the wild type ESXF and ESXE,
yeah.
right? It is that data mapped onto this. So I don't understand that.
No, I
Like in vivo just means that that's probably what's happening, right? Like that's something that creates the phenomena that they've been studying. Everything else does not.
yeah. I mean, I guess part of this is because I'm not because usually with heat maps it's like a three-dimensional graph where, and I can't remember what the like third dimension they're showing, what the red indicates and what the blue indicates. Uh,
Oh, right.
so
Well, let's see.
that
I actually don't remember that either. It had to do with smoothing. We can quickly look it up, right? That's not bad.
yeah, we can look it up quickly. We're gonna read a paper in real time for you.
No, I mean
Yeah,
that's partially what we're doing. Okay, contour map solvation value obtained. And I want to look at how they made it in the methods. uh, Man, maybe I didn't. I felt like I had it at my fingertips before, but I don't see it now that we're here. Oh, because maybe it's in the. No. Okay, I guess I don't know where it is anymore.
yeah, it's
I could have sworn that there was a methods block because I remember reading that they made it using some tool and it was like.
yeah. Because I guess like the difficulty of like scientific papers trying to make like complicated kind of concepts more easier to understand, and sometimes that uh, there are some things that are just much harder to express. And
Yeah.
so I'm not sure what the idea there, but the I guess the issue here is like usually when we get a heat map graph like this, there would be a key that would indicate like what red means, what the green means, what the blue means, and we're not getting that here,
Yeah,
which is
we definitely are not.
which is kind of irritating.
This is your
'Cause
editor side coming out. You're right. You're
yeah,
like, this is unreadable.
this is basically my editor side. This is the point when at like the production phase we go back like, okay, you have a graph with three axes and you're just not listing the third axis. What does
Uh-huh.
red mean? Why don't that could mean anything really? So
Yeah. Okay.
yeah, I
I
need
know. I can't find it. It's probably in the discussion that I read it, and now that's eluding me.
yeah I
But
anyway
again, I don't think I'm not taking these last three figures. Like, I think it's, to me, it just shows when you break it, it breaks.
yeah
Like, and it doesn't. I don't think it should be telling us too much more about this scenario because again, there's going to be other factors to these proteins coming together when they're actually in like a certain condition. So, yeah, I don't know.
mm,
This is yeah.
yeah I mean uh yeah I
this
spent too
is
much
this
time
is
trying to
this is
decipher
this
this.
is abstract biochemical characterization,
Mm,
and the thing is, at the end of the day, that can only be used to help us figure out what is in vivo, right? And
mm,
and we have to be able to match it to conditions that are out there, right? Or at least um speak to like the conformation. Like, I think what they're getting at is this conformation, the pentameric one that they find, it's an important one for the function of this pore.
yes.
Yeah, and it is a pore. I think that's the other thing that they've
Yeah.
shown us. It is a pore.
Yeah, that's an important finding.
Yeah, but
Important
like,
finding.
there's a lot of questions that you ask in this field. Like, what's next, right? Would be like, oh, so like, well, let's just move on. Let's look at like
Yeah,
the figures.
let's move on to like the lab's model of SXE and SXF.
Yeah.
Um, yeah.
So it's we know that it's made. We know that they come together, and that if there only one of them is made, then they won't come together and they won't get out. They The WXG motif is important for this, right? And once it gets into the membrane, then these other effectors can come out, right? Like CPNT.
mm,
And presumably that relates to this whole life cycle of TB, where it lives inside of the things and eventually wants to kill everything and get out. So
yeah.
that's what they've been able to that's what they've advanced here. And I think the biggest thing is really taking these WXG 100 motifs, right, this whole family of proteins, and showing that it is possible for them to make a pore, right? And then that's big. That
Yeah.
it's possible, right? Because
Uh,
now we can start asking questions like, how exactly does it make this pore? They started the story, but they really didn't finish it at all. In terms
but
of how the events that happen. um, but yeah, it's this is the discovery, right? This is a
yeah.
very much a discovery paper.
And I think the important because you highlighted right at the beginning that type 7 secretion systems that you can see how it goes on the inner membrane, but the outer membrane that was still very much a mystery. And this is a protein that lives on the outer membrane,
Yes.
So that's quite an
Yes.
important like step in the chain.
Yes. And they spoke in the discussion about a controversy in the field that came out recently
Mm.
that was talking about ESAT6 and how they think that it doesn't make pores in the membrane as they thought it did because they used detergents or something. It was like a detergent contamination or something like that. And so like they highlight that this what they found here is not susceptible to that criticism because they did not purify their proteins in that way, right? Like they were very careful to show they seem very aware that if they wanted to show pore activity through the methods that they used, that they took care in their purifications to make sure that they could and not confuse people. Confuse the
Yeah.
literature.
So I think that
um,
brings us to yeah.
I want to go to that supplemental figure that I have at the end. I think there should be one more slide.
Okay, hold up.
Oh, is it?
Yep, there is one more slide. Yes.
Yeah, and I think this is a good place to just like peel off the sort of the discussion about like why are they going like why biochemistry to get at this question,
Mm.
right? Like there are other proteins, there are other pore forming proteins, and I could have put this in the introduction too, but I kind of didn't want to bog us down. I wanted to tell more the story of type 7 secretion, but like really the pore forming proteins are like a huge family of proteins even in our cells that they aren't even toxins. Like we need pores, you have to get things in and out of cells, like transporters, right? You need gates, you have to get in
Mm.
and out. And pores are also great toxins because they can go and they can puncture holes and things that you want holes in so you can get stuff out or you can put stuff in. And inside of pore forming literature, right, like there are steps to making the pore, right? Like what is the first protein that gets in? How does that tip get into the membrane, right?
Right.
And then when it gets in the membrane, how does it then find the other things that got inserted into the membrane? Do they oligomerize first and then they get inserted, or do they get in and then they oligomerize? Right? All of these are very important questions about mechanism of the pores that have been found in other pore systems, but not necessarily in this one. And
Mm.
so what I see sort of that ending biochemistry, right? I want to relate it back to that horribly that graph that didn't have the right legend on it, is that like they're trying to build up that body of knowledge so that we can relate it to other like biochemical stories that we have of how pores work. And hoping to relate it there will then give us an understanding of how this one works and maybe generate a testable hypothesis that then we can use to look at deeper into the function of ESX, Essex, F, and E.
Right. Yeah. Uh, so I don't really have much to add because I think that you're the real expert on the on this like protein
Oh,
side of things. So,
yeah.
um,
I mean, like maybe just to speak generally about like right at the end of the day, all of this is about pathogenesis, right? Like
right.
this is a type of pathogenesis paper that we looked at, and we've been looking at over this past time of looking at SARS-CoV-2 stuff, right? We've looked at a lot of impacts into the clinical side.
Mm.
So like maybe a question, just a quick discussion to wrap it up is like, so how do papers like this that talk about pathogenesis relate to like disease itself, right? What's the way that this stuff gets linked up?
Well, I mean, the obvious one is saying, oh yeah, treatments. We're gonna find drugs that will fit into these, and maybe we can find one that can go make the jump intracellularly into macrophages to attack these pores directly. But
Totally.
I mean, another thing could be to because I know with TB specifically, it goes dormant for a time. So there's lots of times when it's not producing these proteins, and maybe these could be biomarkers signals to say that oh, this is when the granulomas are suddenly going to be active or not. So,
Sure.
um, but I guess also like looking at this from
Oh,
the perspective of molecular machines working to do molecular machine stuff, understanding how these work can mean that you can develop things that mimic that use the knowledge from here to develop your own machines to do things that you want to do.
mm-hmm.
So let's say if I want to design a molecule that will be inside the membrane or that will help a drug get inside somewhere, then maybe these kind of studies help build on that knowledge.
Totally. Um, Minimal just wrote in our chat. What's the title or episode of the soluble ACE2 decoy episode? The so the ACE2 decoy episode is this one. De novo design of potent and resilient H2 decoys to neutralize SARS-CoV-2. But I suspect that Oh, I just realized that my Zotero is not open on the same computer that I have the chat. Okay, so I can't paste that in. But the other thing is that, but I think that the one where they used tritium to actually look at salvation, that was one with antibodies. Do you
Oh,
remember that one,
I
Faz?
remember that one. I
Where we
think because
looked
that
at...
was the one where I did the analogy that's like fried chicken, where I deep fry the antibody, and I think that was I feel like that was a
Yes, that's
Regeneron one.
right.
Yeah.
The
Uh,
Regeneron
I,
episode.
f-
Okay,
I feel
so
like
that's going to be monoclonal. That's going to be antibody cocktail of SARS-CoV-2 spike protein prevents rapid mutational escape seen with individual antibodies.
yeah, I think that's the right one. Although I mean, I could be because at this moment we've done so many antibody papers. I feel like they've all meshed
Okay.
up in my
Well,
mind.
hopefully that's enough to send them on a journey of searching.
Yeah, I mean,
And
I'll
I can drop a response. We can drop a response in the comments.
yeah. But thank you for commenting, Minimal. It's always great to have people live commenting while we're here so we can respond to you in real time.
Yeah. Okay, so I think that brings us to the end. Yeah, join us next week for our news week where we're going to survey papers to find something that we want to cover in detail the following week.
Yeah. And sorry, I've got my blurb. Yeah, and thank you so much for listening. And let me see what because there's always some stuff that I need to like things like every YouTuber needs to say like oh like subscribe and I didn't write it in my notes. So
Yeah,
okay,
absolutely.
it's
I mean, no,
yeah. So
we can go casually too. I don't mind.
yeah, sorry. Yeah, no, we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified by it, it's possible that we didn't get everything right. So science is about thinking critically. So ask the right question and asking about the right question. So if you have any questions or corrections, please let us know in the comments.
Yeah. Again, reach out to us over Twitter using the hashtag #MicroTWJC. We think that peer review is this process that we can all participate in, so that's why we want people to ask us questions. We hope that you had a good time listening to us ramble about microbiology, and yeah, it's been a pleasure chatting with you.
It's been a
Buzz.
pleasure chatting with you, Danny.
All right, tune in next week for more microbiology content.
All right, bye.